Hello, all, and welcome. This is Mrs. Bryant at approximately 12:10 a.m. So I'm a little sleep deprived. I apologize I couldn't be here again today. Yesterday, my daughter was ill, and she somehow passed the bug to me, and now I'm not feeling great. So hopefully, I will be back Wednesday, which is tomorrow for you guys. But for now, make sure you've submitted your diffusion homework or not your diffusion homework, your 5.2 homework, and then complete the following: this ed puzzle on your notes on osmosis and the two following ed puzzles on cell transport and homeostasis. And those are the main requirements of the day. Now, I apologize again. As of yesterday, I still don't have my packet with me, so I don't know the exact order of my pages or the page number, so I apologize. Hopefully you were able to find the diffusion page and the page on the flowchart, so you can fill in those notes. None of you emailed me otherwise, so I'm going to assume that's the case. Please expect a ticket in the door when you guys come in tomorrow on the stuff from today. I'm going to throw a couple questions in here right now from your homework. Hopefully you did your notes. So now I want you to flip to the next page in your section. I might be bouncing back and forth. I apologize if I am. And I'm also not showing my face because I want to make sure you guys can see all of your screen, all of the stuff on the slides. So yesterday you were looking at various types of transport focusing on diffusion. Right now we're going to be primarily looking at passive transport. So diffusion, osmosis, and facilitated diffusion. If you remember, diffusion is when you have either nonpolar or small molecules moving from a high to low concentration or down the gradient. It's kind of like going down a slide, spread out. I want you to imagine like somebody spraying perfume and it slowly going everywhere. You won't always smell perfume right where it's been sprayed. It'll start to move to other parts, and eventually you won't smell it at all. Osmosis, which is the movement of water in that similar direction of high to low, spreading out. It doesn't really require any energy, just like diffusion. Any passive transport, it's passive, no energy required. And then finally, facilitated diffusion, where you actually need help to get the molecules across. You still don't need energy, but in order to get the larger and polar things across, you might need a protein, like those channel proteins we talked about before break, where it's just like an open passage in the membrane to allow molecules through, just like when we did the bubble lab and you got to see the opening with that little piece of string. So let's focus on osmosis today. So you read on notes all about diffusion, so let's look at osmosis today. Oh, and tomorrow also be prepared for a lab, because we will be starting a lab on diffusion tomorrow. And you'll be practicing diffusion and osmosis problems as well. So now, like I said, I don't know if I'm in the right order, and I apologize. I didn't grab a packet before I left for break, and I didn't anticipate being out. So I apologize if this is out of order, but find this page in your notes and start to copy it down as I'm talking about it. I want you to include the arrows and the red words to help you understand. So there are three types of solutions a cell can sit in. Each solution will affect a cell based on how much liquid versus how much dissolved particles. So the solvent and the solute. Remember that a solute is the stuff that's being dissolved, and a solvent is the stuff doing the dissolving. So the first type of solution a cell can sit in is a hypertonic. Hyper means above. Think of somebody who's had a lot of sugar. And actually, this is quite the case in the environment as well. In the environment, there's going to be a high amount of solute and a lower percentage of water compared to the cell. So it's like you've dunked your cell in sugar water. Because of that, the particles on the outside the particles of sugar, salt, or whatever other solute you have in there are going to want to move into the cell, and the water will move out to try and balance things out. So you're going to have a rush of particles in and out. So the water will come out and the salt and sugar will come in. That's why it's hypertonic. You've put a cell in something that's like souped up in sugar or salt or something like that, and so the particles are going to want to go in and the water is going to rush out. In a plant, this is going to cause a plant cell to buckle Those cell walls are actually a nice extra layer of stability and protection. They will eventually collapse, but they buckle, and we call this plasmolized. In animal cells, they will shrivel up. This is like if you go to the beach and you drink salt water, the cells that the salt water comes in contact with will shrivel. This is why also swishing salt water, if you have like a mouth sore or a swelling in your throat, like if your throat's swollen, they tell you to swish salt water. That's because that salt will enter your cells and will force water to come out of your cells, reducing the swelling and actually easing the pain, which is great. The second type of solution is called hypertonic. Hypotonic, I apologize. The names are very similar. Please make sure you keep them straight. Hypo means below, like hypothermia. You can also go into hyperthermia, where your temperature gets too high, but hypothermia, like when you get too cold. But hypo means below. And this is when the environment on the outside of the cell actually has a much lower salt, sugar, or whatever solute is being used consistency compared to the water. So it's kind of like you dunked the cell in pure water, or like very watered down material. So what's going to happen is the actual solutes in the cell are going to leave, and then the water is going to rush in. Good in some cases, bad in others. This is really good for plant cells. Plants actually really, really like this. This is actually what gives them stability. We call a plant cell that is filled with water in such a way turgid or turgid. This is ideal for plant cells. They like that extra water. This is what allows them to stand upright. If you notice, if a plant doesn't get enough water, it starts to wilt. But if it gets a little extra, it will force it to stand up. We, on the other hand, don't do very well this way. If we get too much water, we go through something called lyse, which, if you remember from root words in the past, lysis means to break. So if we get too much water, we pop. Not great for us, good for plants. The last type is isotonic. Iso from before meaning equal. Isotonic means there is a perfect equal amount of solute on the outside and inside. So perfect amounts of liquid, perfect amounts of dissolved substance. So there's going to be constant moving. This is what we call dynamic equilibrium. These molecules are not going to stop. Even if they're perfectly in sync, you're going to have the same amount moving in as moving out, and just constantly cycling through at perfect matched levels. Plants, they don't really like this. They're actually needing more water, and that's when you notice, like, if you've been keeping a houseplant, it's starting to droop. This plant cell state is called flaccid. In animal cells, this is great. This is actually perfect for us. We love this. It is ideal. Now, I do want to clarify, this does not mean that your solute and your solvent are 50-50 on both sides. It could be your solvent is 75 on the outside and 75 on the inside, and your solute is 25 on the outside and 25 on the inside. The solvents must match, the solutes must match. They don't have to be perfectly equal 50-50, 50-50. It doesn't work for all things at all. But this is actually perfect for us. Now, let's talk about osmoregulation, which is the regulation of water. And what happens when bad things happen to us in these sort of situations. So, in us, and other organisms with kidneys, our kidneys actually filter out the extra solute from the blood and the solvent and excrete it as urine. So they'll take out the extra particles that we don't need, or the extra water. Your kidneys and your urine are actually a great sign of how your homeostasis is doing. If you look at the color of your pee, the lighter it is, the more water you are excreting because you have too much water in your system. If it's too light, you've got way too much water in your system. If your pee is dark, you're not getting enough water. And you can die from drinking too much water. There was actually this horrible, terrible case. It's very sad. Called the Hold Your Wee for a Wee contest. This was in Sacramento, California in 2007. This lady's name was Jennifer Strong. She was about 28 years old. She had 2 sons and an 11-month-old daughter, and she died from something called water intoxication. She enrolled in a contest, it was a radio contest, that you needed to drink as much water as possible without going to the bathroom. Each contestant was given 8-ounce bottles every 15 minutes, which is about half of what you normally think of when you think of a water bottle. And after 5 bottles, you were given a normal water bottle worth every 15 minutes, and the goal was to get you to drink 2 gallons in 3 hours. And that's what she did. Unfortunately, she died. Their legal department stated, well, it's fine, the body's mostly water. What could go wrong? How could somebody die from this? She went through something called hyponatremia, which symptoms include headache, confusion, muscle spasms, seizures, and then death, which is when her sodium actually drops really, really low because there's way too much water and there's not enough sodium getting to the cells. It's very deadly, and we'll explain step by step what happened there. Her family ended up winning a gigantic legal battle, which was very sad because this happened all around Christmas, and she was trying to win this Wii for her kids. But the family ended up getting about $16.5 million out of the lawsuit because the radio show never went through their legal department about this. So what happened was her cells ended up being immersed in a hypotonic environment where the solute was low and the water was high. Because she couldn't pee, her body couldn't get rid of the excess water, maintaining homeostasis. The water rushed into her cells, going from high to low. The water diluted the sodium, and the cells burst. And they died faster than they could be replaced, and she died. So that sucks. And you can't do it. Don't do it. The amount of water she drank in under 3 hours is the amount a pregnant woman should drink in a day. So don't do it. Or some athletes might drink in a day, but don't drink that much water without going to the bathroom. You will die. So in plants, plants actually prefer hypotonic. And this is because the extra pressure, which we call turgor pressure, pushes on the cell walls, keeping the wall structurally stable and the whole cell kind of nicely in place and rigid. And it's great. When a plant cell loses too much water, it goes through plasmosis, and that's usually due to a hypertonic, and you see this shrinking happening. It'll pull away from the cell wall, and the plant will not be structurally stable. So turgor pressure is very needed. You need high turgor pressure for plants to do well. And finally, the last thing I'm talking about is another way plants regulate, and that's through a special vacuole that they have in some of their cells called a contractile vacuole. And this is really cool. It kind of acts like a pump. This is really, really important for things that live in like ponds and lakes where their environment is naturally hypotonic. And so plants eventually will undergo too much pressure and they will burst. Their walls can only hold so much. They like some pressure, but too much is too much. And so they have these neat little contractile vacuoles that will pump water out when there's too much to try and keep up that regulation. This usually is in like single-cell prokaryotes or protista or fungus that live in the water, that live in sort of like a very freshwater environment. But it will literally act as a pump and just pump out the excess water. All right, that's it for me on osmosis. I'm stopping there. I know I've talked for way too long, but I hope the story was interesting, and I hope you learned a bit about osmosis and the different types of solutions. So now watch the 2 other ed puzzles, and you're done. You can continue working on your homework. I should be back tomorrow. Be prepared for a lab.